Welcome to Study Loop.
For this question, we are looking at histone modification in newborns. So in this case, we are looking at a newborn rat with a GR gene. For this question, it's not particularly relevant that we know what the GR gene does. However, we do know that it is a newborn. However, for curiosity, the GR receptor is the glucocorticoid receptor inside of rats. So it plays a role in hormone signaling and in some reproductive functions. However, inside newborns, we have to consider what state the histones are going to be found in. And this can be in one of two forms. It can be in either the methylated form, with the methyl group attached to the histone proteins, or they can be found in the acetylated form, where the histone proteins will be farther apart. And of course, the histones play the role of winding and coiling the DNA around it to help condense and localize it to the nucleus. So in the methyl group, these are highly bound and there is very little space between our histone proteins. Whereas in the acetylated form, we have lots of space between the histones and they're less tightly bound. What's important about this is there are going to be genes inside those open spaces of the DNA. However, in the methylated form, this is very tight and doesn't allow room for polymerases or other transcription factors to bind. So in the methylated form, this is going to be sort of deactivated DNA, you could say, where transcription and translation are not ongoing. Whereas in the acetylated form, there's plenty of space for your polymerases and transcription factors to bind to create that mRNA molecule. So the acetylated form is more of the activated form. So considering that this is a newborn, you can imagine that most of their DNA functions are going to be inactive or in a state of sleep because they are not particularly exposed to the environment or the surroundings around them. And they have been given suitable nutrients through the mother, so they shouldn't have had much need to regulate and open most of their genes in this acetylated group, and they have been in more of a senescent or more sleeping state. So here, the best answer choice for this question is going to be A, that you would expect most of the histones would be methylated and sort of inactive at this point, because they are newborns and they have not really needed to produce their own proteins for their own functions. So here you should pick answer choice A.